We may know much about the brains, yet even more remains to be discovered. Researchers don't even know precisely how many different types of neurons there are in the brain, much less how they are connected. So, introducing to you the first of its kind, EyeWire. For my citizen science project, I chose to participate in the citizen science game called EyeWire. EyeWire's mission since 2012 is to map neurons. They work to solve one of the biggest bottlenecks in neuroscience by pairing the superior human insight of gaming with AI, which is artificial intelligence, to reconstruct neural circuits. Why is EyeWire's mission so important? Well, scientists know that the human brain is made of billions of big branch cells called neurons. These all work in tandem, millions of cells connected through timed iconic impulses, firing in the vast waves that impact the chemical environment of the brain itself. Your brain is connected, relaying signals whether you're reading an article, playing a game, or simply sleeping. Citizen science turns the act of mapping neurons into a 3D puzzle. Ultimately, it brings us closer to understanding the brain. So getting started, EyeWire has you create an account to keep track of your progress while mapping neurons. That's right, with no neuroscience experience, people like you and I all around the world can learn to solve these puzzles. So how exactly do we map neurons? Well, have you ever wondered how scientists are able to measure and study tiny cells in the brain? Well, scientists use many techniques that each provide different types of scales of data, which combine to yield rich information about the brain. Here on EyeWire, we take a walk through mapping circuits at Synnable Revolution, which enables us to chart the connections between neurons, and we begin to model the brain. This field of neuroscience is called connectomics. This process begins by recording firing patterns of neurons. Researchers can record activity over time from tens of thousands of neurons at a single cell precision using 2-photonic microscopy. Neurons in this poster, shown on the right, come from a visual cortex and respond to the specific aspects of sight. This is the first stage in understanding how visual information is processed in the brain and knowing which cells respond to which stimuli. Secondly, they reconstruct the same neurons, starting by imaging a volume of brains with an electronic microscope. This yields pictures roughly 15 times higher resolution than those taken on a typical iPhone or Android device. Using several types of AI combined with human insight, 3D maps of neurons can be reconstructed and presented as a network. For the last 2 months, I spent 2 to 3 hours a week on this project. Here is a live video of me playing the game. And here on the right is where I manually kept my data. Every day that I logged in, I put it in a Google Sheet. On the left-hand side, you'll see an interactive cube. This is a tiny volume of a real brain, and a neuron branch has grown through it. The challenge in EyeWire is to map that branch from one side of the cube to another. This is done by clicking colors inside the lines. As I scroll through using my arrow keys, I am looking for parts that need to be colored in, and with one click, I can add a piece of neuron branch. The plane on the left side moves through the cube on the right, and you can see the cross sections of the branch that are being matted together. While looking for places to color inside the gray lines, I am mapping the brain. The gray lines are the edges of the branch of a neuron that were stained red on purpose to be noticeable for users. To successfully complete a cube, I want to make sure that all the branches are touching the edges of the cube to make sure there isn't no jagged edges, because neurons don't have jagged edges. This can be checked by scrolling all the way through one final time to double check. When I'm done finding all the pieces, that I thought went with this neuron. I clicked I'm finished located at the bottom of the screen to submit this cube. From there, I'm awarded points based on how long it took me and the volume added that was agreed with other players. So when you get back to the home screen, you can take a look at the leaderboard and see what each person has colored in and where they're from and their score. You can also click on your own profile and look at your points and your cubes that you have completed. You can also check out your accuracy and see how far along you are. That was my citizen science project. Thank you all for watching. Here's my works cited. I hope that you enjoyed and that you learned a little bit more about neuroscience and how scientists map the brain.